So I'll be your host for today's webinar, and it's my great pleasure to introduce our speaker for today as well. We have Dr. Sean Tomlinson, who is a researcher in Perth, also here in Australia. And I've had the great pleasure of writing a lot with Sean over the past several years on a lot of really important projects, and also for our Conservation Physiology in Action section, which is in the journal Conservation Physiology. Sean is a conservation biologist, as you can see from the title, and he works on ecological restoration, wildlife management, and he's worked in an array of different institutions from the private sector, government, and of course, universities. And he's devoted a lot of his career to developing empirical evidence-based approaches for conservation. So we're really excited to have him join us for today's webinar. So without further ado, it's my honor to introduce Dr. Sean Tomlinson as our speaker for today. Sean, you have our attention.
Thanks very much, Jody. So I guess just springboarding off what Jody said, I'd probably have to start by acknowledging that a lot of this work was, this research work I'm going to talk about was conducted in the traditional lands of the Noongar people in the southwest WA, and just pay my respects to past elders, past, present, and emerging, and their traditional custodianship of the land. I guess the themes that I'm gonna riff off here are actually covered in a paper that I wrote with Jody, Kevin Holteen, and Steve Cook, called Crossing Boundaries in Conservation Physiology. And what I'm going to play around with today is kind of my own personal journey in taking principles of thermal biology that I learned during my PhD, and then kind of carrying them across different taxonomic boundaries and pulling together different themes and stresses to really see how transportable those themes and concepts of thermal biology prove to be. And it all started when I was playing around with these guys, thin-eyed wasps, and the orchids that they pollinate, where each orchid has a specific relationship with a specific species of wasp. But we'll get to that. The structure of this seminar is going to kind of run in 5 themes. You can see them outlined at the top there. I'll begin with just some general conceptual ideas about how I approach research in general, some stuff about the principles of thermal biology, although I'm almost embarrassed to cover some of that off looking at the list of audience members I've got here. There's a fairly imposing list of names there. And once I've covered off the theoretical concepts, I'll look at some stuff about insect physiology, moving into plant physiology, and then looking at where I think we might be going in the future. And the kind of underpinning ideas, the rationale behind all of my research was a set of ideas that I pulled together in 2014, which are about how you take physiological data and put them in a context that really allows you to drive and guide conservation outcomes. And I broke the concept down into 2 models. Essentially, what you can see here is the environment model. You have to have some kind of spatiotemporal understanding of what the environment is, and you need to break that down into the environment from the perspective of the organism you're working with. So it might be temperature, it might be soil temperature, soil water content was something that I had to learn all about when I started working with plants. pH, if you're looking at marine organisms, for example, I can see how that might be important. Once you've got that spatial template or spatiotemporal template, you then also need to have a model of how your organism works. And this is the physiological kind of understanding. This is a true mechanistic understanding as opposed to a correlative understanding of what conditions are like where the organism occurs. Again, we'll come to comparisons of what those two models mean later in the seminar. But in this circumstance, you might measure a physiological trait, be it metabolic rate, which I measure a lot, running speed, germination rate, and you can kind of build these physiological performance landscapes, which give you an understanding of how does the organism respond in a really mechanistic and specific way to the challenges that it encounters in its environment. And once you have those two, you can solve these two mathematical models together. You can make them interact, and this can give you some really interesting insights into what's driving or constraining persistence or reproduction or performance, to give you a realized ecological distribution, an understanding of the mechanistic underpinnings of the realized niche. So that's kind of the broad brushstrokes of how I bring my ideas together. And over the course of my career, I started out working with in my PhD with mammals, working with Phil Withers. And since then, I've done stuff on insects and insect pollination and rare plants. Rare plants led me to soil fungi. And I've also been lucky enough to explore some of these ideas in marine ecosystems. What I'm going to stick to today is just my transition from insect physiology into plant physiology, because the thermal biology translates quite well, and I think it's quite a nice example of really pushing those taxonomic boundaries, because botanists and zoologists, perhaps more than any other groups of biologists, tend to think quite differently from each other. I'll leave it to the audience to decide who thinks right. I am Switzerland when it comes to those kinds of conversations. So the principles of thermal tolerance. Forgive me if you are already well versed in this and teach basically these slides to your undergrads. But the principles are that metabolic rate should increase as temperature increases, at least as far as ectotherms are concerned, because thermal performance and metabolic rate in particular are essentially defined by chemical kinetics. Chemical reactions have more energy as the temperature increases. Chemical reactions go faster, therefore metabolic rate is a chemical reaction. As the temperature increases, it should go faster. Metabolic rate is also catalyzed by enzymes, and enzymes are proteins, and proteins all denature once they reach a certain temperature threshold. They can only catalyze reactions up to a certain point, and then it all falls apart. So once you exceed that threshold of protein thermal tolerance, the whole mechanic of how metabolic rate is catalyzed begins to collapse, and metabolic rate starts to decline. And so you get this kind of idea that metabolic rate is going up as temperature increases, but eventually it starts coming down, and there should be a peak in the middle that provides some kind of meaningful threshold. And as a piece of data to provide evidence for this, you can see that when you measure metabolic rate. So this is where I began my journey working with Ryan Phillips, who was at the time with Kings Park. And he was interested in these thinine wasps because he was actually interested in how these wasps pollinate orchids. So he wanted to understand what is the thermal tolerance of the wasps, and does that influence how effective they are as pollinators. And we did a whole bunch of measurements on a whole bunch of different species, one of which you can see here. And metabolic rate does indeed go up it reaches a peak and then starts coming down. And so the theory runs that at that peak, at that temperature of about 35 degrees, the organism is operating in its absolute peak efficiency. It's moving as rapidly as it can. It's foraging as rapidly as it can. It's burning energy at its sort of peak capacity. This is its optimal thermal operative temperature. It's T-opt. And traditionally, you would just linearize the data as I've shown here. This is precisely the analysis we did. We applied a broken stick regression, and the broken stick estimated where the T-opt was, because the T-opt is that breaking point. And this is fine, and we got it published, and we were quite chuffed with ourselves because we were both PhD students at the time. So this was sort of our first foray into independent research, independent of our supervisors, I should say. But it always kind of bothered me that you can see, even from these points, that that increase isn't a straight line. And it bothered me that I couldn't think how to get a better estimate of this that captured that curve, because I couldn't think of a way to do a non-linear broken stick regression. It didn't exist at the time. And so I spent a few years thinking about this and playing around with the ideas, because it actually makes perfect sense that these relationships aren't going to be linear. We know for a fact that very, very few things in nature happen on linear scales. And so I came up with this concept that you could probably, if you broke down the components of how thermal performance works into increasing performance and this enzymatic kind of threshold cap, you could break it down into a theoretical minimum performance, because, unless an organism is dead, it's doing something. In terms of metabolic rate, it has a basal metabolic rate or a resting metabolic rate. And you have a theoretical maximum, you have a peak metabolic rate, beyond which the machinery of the organism just can't generate or process energy any faster. And so if you've got a maximum and a minimum and a nonlinear curvy kind of response in the middle, you get a logistic function. So I came up with the concept that the performance increase is probably logistic. That's the easiest thing to fit, an S-shaped curve. And enzyme activity would theoretically be perfectly stable until you reach this threshold. And then you'd start to see it gradually declining, because it's not like all of the enzymes denature all at once. You don't have this kind of sudden crash. You have a tapering off that gets steeper and steeper as more and more of the enzyme pool available, I guess, breaks down and denatures. And when you combine the two, you get this really characteristic unimodal hump-shaped curve, which is how thermal performance curves have been described ever since Raymond Huey started throwing these ideas around, and probably even earlier. Huey is just the name that sticks in my head. Angeletta, another one who's described these as unimodal hump-shaped curves. And so by combining a logistic function and an exponential decline relating to enzyme activity, you can build mathematical functions. So I proposed this mathematical function that describes these unimodal hump-shaped thermal performance curves. And that function has a bunch of really important parameters associated with it. So you can capture In this case, WF is the peak performance, WO is the minimum performance. You can capture thermal optimum, T-opt, because that is the point where the curve turns. You can capture Tmax, so that's the point where the organism, where performance equals 0, the organism has died. And you can interpolate a couple of interesting thresholds from this as well, as kind of I termed them TD lower and TD upper. They were sort of 90th percentile performance thresholds, just to kind of provide a boundary of this is kind of where the organism might want to operate, raising the question of, is T-opt actually T-opt, which has been bandied around in the literature for a while. So that's the principle behind how I kind of got from a linear understanding of thermal performance to a nonlinear understanding. And this nonlinear understanding is really important because it provides you one mathematical equation that you can then solve across that spatiotemporal template that I was talking about before. You know, if you have a series of temperatures across a landscape, you can then theoretically solve this equation across that landscape and say, and understand how performance of the organism will change, spatially and temporally. And I was really, really proud of this equation right up until I started working with plants and discovered that a pair of botanists had come up with something much simpler and much more effective 10 years before I even thought about it. But that's a story that we'll get to later in the seminar. At the time that I was developing these, what I was really interested in was I was no longer hung up on how wasps pollinate orchids, and I was much more interested in the idea of how do bees in particular interpret a changing landscape, because bees contribute the vast majority of pollination services globally, and certainly in the southwest of WA, where I was working at the time I did this. And bees fulfill, particularly native bees, particularly non-honeybees, non-apis in Australia, fulfill an interesting array of pollination services, because some species fulfill quite a generalist niche and they will pollinate anything and everything. Other species fulfill quite specialized niches. And so if you're looking to manage a landscape or restore a landscape, you might not get natural recruitment from some of the plants that you're trying to restore if their specific pollinator isn't there. But you also want to make sure you're capturing species that, or you're accommodating species that fulfill a broad niche, because you want to get as many species back as you can. So bees were a really interesting thing to play around with. And the two species that we looked at in this particular study were apis honeybees, shown here in the orange boxes, and a thing called the blue-banded bee, Amygdala chlorocyanea, which is the lower image here shown, and the blue points in these plots. And we used those nonlinear thermal performance equations that I showed in the previous slide to characterize how the metabolic rate of these two species responded to temperature early in the spring season and late in the spring season, because they both have a flying season that starts about now, actually about October, and they'll fly all through the summer until maybe March or April. And what I want you to take away from this particular slide is that early in the season, the two species are fairly similar. They've got moderately similar peaks of maybe 6 mils of CO2 per gram per hour for Amygilla, and maybe 7 for the honeybee. And their peaks are, their T-opt are both in the low 30s. Their upper thermal tolerance is maybe 35 to 37 degrees for both species. But this is early in the season. This is about now, about September or October, when environmental temperatures, I mean, today in Perth it's 38 degrees, and for this time of the year that's absolutely sweltering. So for early in the season, they don't need particularly to be adapted to high temperatures, but summer in Perth can get to temperatures exceeding 40, 45 degrees. And when we measured their metabolic rates and their thermal tolerances later in the season, again here the orange boxes are honeybees and the blue circles are blue banded bees. You can see that the takeaway message here is that both species are able to accommodate a higher thermal tolerance later in the season. For blue-banded bees, their peak is at about 40 degrees. For honeybees, it's about 45. So honeybees are slightly more thermally tolerant. But the comparison that really struck me here is that for honeybees, with the 2 dashed curves, the energy requirement when they're operating at their peak doesn't shift very much. It's 7 to 8 mils of CO2 per gram per hour, regardless of season. But Amygilla, they, for whatever reason, as they become more thermally tolerant, their energy requirements really accelerated. So this has pretty profound implications, because Amygilla are really important in Western Australian ecosystems because they form really specialized pollination associations. They're a buzz pollinator, and so the way that they access the nectary in the plants that they service is they fly up and they hang on to the underside of the plant, and they vibrate their wing muscles at a harmonic frequency that causes the anthers to open up, so that the flower is closed and inaccessible until it's hit with a harmonic frequency or a harmony of the right frequency. And then the anthers open up, the bee can access the nectary, and as the anthers open up, they shower the bee with pollen. So the only way you can get pollen transfer from plant to plant is to either use a tuning fork, which will again release pollen from the plant, or to have a bee that can vibrate its wings at the precise frequency. It has to be one of these Amygilla. So for a fairly large proportion of the plants in the southwest, they don't get pollinated if they're not being pollinated by a bee that buzzes at the right frequency. And this has some pretty profound conservation implications, because the landscape we were working in at the time, if you look at the regional map here, panel A, you can kind of see a red splodge just north of Perth. This was all pine plantation put in the 1960s. And by the turn of the century, the pines were being sequentially removed, and it left 152 square kilometres of restoration space, which is what's shown in the study area panel B. All of that pale green area is ex-pine plantation. And the whole area has been developed in a sort of myriad of ways, with some urban development and some farming and all kinds of things. But it was the pine plantations that we were focused on, because once the pine plantations came out, what was left was just kind of bare sand plain, with the occasional little remnant patch of Banksia woodland spread across the whole landscape. And so you've got these isolated little patches, some of which is being and some of the landscape is being restored, but obviously it's not all being restored all at once. And the question was, how do you encourage the bees to move outside their little patches and into these restoration landscapes when the surrounding landscape is now suddenly much more exposed than they're used to? There's no overstory. It's quite hot. It's quite dry. There's no cover. There's certainly no nectar resources. And so our question was, what is it that's limiting the capacity of these bees to invade restoration sites? Was it the fact that the sites were just too hot? Did it exceed their thermal tolerance limit? Or is it the fact that they could tolerate those landscapes in terms of thresholds, in terms of absolute capability, but their energetic budget was being absolutely blown by the changed temperatures? And so this was our spatial template. We generated topoclimatic models that gave us temperature across a gridded landscape that you can see here. And each grid cell was about 25 meters by 25 meters. And once we had an idea of what the temperature was doing across that landscape, all we had to do was solve those thermal performance equations to understand how close to their threshold were each of these species, and B, what was the energetic expenditure for a bee in that grid cell for an hour. And that's what I'm showing here. The top panel is the honeybee, the bottom panel is the blue-banded bee. The panel on the left is just thermal suitability, showing how much of the landscape is approaching that, or how close that landscape approaches their optimum thermal temperature. Their thermal maximum. And for most of the landscape, they're within their thermal maximum. There's not a lot of the landscape that exceeds the thermal maximum. So the takeaway that we had from this is, okay, it's not temperature that's limiting these bees. They can move across the landscape and not just die from heat stress. But we also calculated out the energetics of being in any of these 25 by 25 meter grid cells for an hour. And in parts of the landscape, the blue-banded bees are spending 16 odd mils of CO2 per gram, mils of CO2 per hour that they're spending in those grid cells. Honeybees are spending slightly more, up to 20. And this is modeled for an October day. Now, no one really understands what does 16 mils of CO2 equate to. And the good thing about CO2 is that you can convert it to energetics. And when you do that, you realize that blue-banded bees in parts of this landscape are chewing through the energetic equivalent of about 3.5 cans of Coke every hour they're out there. So when you think in terms of a bee that weighs maybe 3/4 of a gram, having to go through 3.5 cans of Coke, you suddenly have this idea that we need to put a lot more energy resources back into these landscapes to get native pollinators back in there. And this is for spring. For summer, we know that it's even higher because of the way those thermal performance curves changed through the season. So this was kind of the take-home message here, is that perhaps restoration in these landscapes needs to think less about replicating nature and more about providing artificially high volumes of nectar just to reinstate pollination services. Because if those pollination services aren't there, there's going to be no natural recruitment, and restoration ultimately will be a kind of effort of ongoing gardening. Now that's fine if bees are the only pollinator, but all of these things are context specific. So when I went and had a look at what do beetle pollinators do in these landscapes, beetles are an entirely different circumstance, and beetles are really understudied in terms of both their physiology and their role as pollinators. And as a case in point, the particular beetle that I've got photographed here, this thing is huge. You know, it's maybe 5 centimeters nose to tail. It weighs nearly 2 grams. It sounds like a low-flying aircraft. The sites where I was catching this are only 30 kilometers from the center of Perth, and it doesn't have a scientific name yet. So this was new to science when I started working on this species. And you'll see from the curves there, they're much more thermally tolerant. These guys fly through the summer and they can tolerate temperatures up into the 40s. They've got a much lower energetic requirement. And as a result, when you solve their thermal performance across the landscape, and they're nocturnal as well, so they face a much lower environmental challenge. And so when you solve their equations across the landscape, they can now access much more of the landscape. There's fewer thermal barriers to them, and they require much less energy to do so. So I guess the caveat that I put on this is that a landscape designed for bees needs to be highly energetic. A landscape designed but a restoration sequence designed around beetles can afford to be less energetic and still capture restoration potential just by switching the focus towards a different pollinator. I think ideally, to guide the restoration of these landscapes, you perhaps want to do both. You want to aim to capture bees because you know you'll get beetles, but you want to think about beetles as well. I didn't have a chance to do very much in the way of restoring these pollination networks because the postdoc I was working on came to an end. And I was faced with a new challenge after that because someone decided that if I'd done work on pollination, maybe I'd be okay as a botanist. This was quite a shock to me because I had not actually done any botany since I was an undergraduate student, but nevertheless, I took on a role at Curtin University after this, principally looking at the restoration of rare plant species in post-mining restoration. And I was fortunate to have a couple of very good colleagues in this space, being Wolfgang Lewandowski at Kings Park and Shane Turner at Curtin University. And what really caught our attention once we started working together was that one of the limitations to restoring plants is obviously getting them to germinate where in the landscape that you've re-engineered for them, that has been restored or is to be restored. And germination is a really interesting life history stage for a plant because the plant's not photosynthesizing, and the seed is basically forced to, I guess, conduct all its activities based on a store of an energetic store that it has inside the seed. So before it can get its cotyledons up and get its leaves out and start photosynthesizing, my I guess thesis statement was that plant seeds aren't really plant seeds. Plant seeds are sort of like small ectotherms. They're kind of ectothermic eggs, which means that they should be faced by similar thermal tolerance problems as an ectotherm. And Shane and Wolf grimaced, told me I was an idiot, and then decided to maybe sit down and see how we could work with this idea. And germination is an interesting process because it's characterized by sigmoidal curves over water stress. So at very low water stress, you have high germination. At higher water stress, water potential in this case, as shown on the x-axis here, you get lower and lower germination, and you have these kind of S-shaped curves with a point of inflection in the middle where 50% of your population germinates. And these sigmoidal germination curves are characterized by 3 mathematical parameters. And at least 2 of those, I suspect, are functional traits, being the slope, the steepness of that germination curve, and also the maximum proportion that can germinate. And so you can get this whole range of germination characteristics across water potentials, depending on what proportion of your seed population will naturally germinate and how rapidly that germination declines in relation to water potential. And so some of those traits have very familiar responses to temperature. We were fortunate enough to have a student who was really interested in taking this on, Subashi, and she looked at a whole bunch of different species of different germination syndromes and tried to quantify the thermal performance of that germination rate parameter. And this is where I stumbled on the Yan and Hunt function. I say stumbled, I think it was Wolf who told me that someone else had come up with a much better idea of this, and this was how it worked. But the Yan and Hunt function is actually a really good thermal performance function. It was originally designed to quantify photosynthesis in corn. And I've since applied it to all kinds of species. I have yet to find another function that outperforms the Yan and Hunt function, no matter what species group you're applying it to. And in this example, Subashi was looking at trying to understand how germination, how thermal performance of germination works for short-range endemic species versus widespread species, because the idea should be that short-range endemic species have they should be stenotherms, right? They should be thermal specialists because they only occur in very specific places. They should have a high peak performance but a very narrow range. And this is precisely what she found when she was looking at the rate of germination in Eucalyptus caesea and Eucalyptus ornata, which are the 2 on the left-hand side, the 2 left panels, versus Salmonifloia and Salubris. Caesea and ornata are habitat specialists. They only grow on rocky outcrops. Salmonifloia and Salubris grow on the kind of sandier, deep soil plains around these rock outcrops. And when we fitted a thermal performance function, we found that, yes, indeed, the thermal specialists have these much tighter thermal performance curves where they germinate much more rapidly at very specific temperatures. Salmonifloia and Salubris can afford to be a bit less selective about the temperatures that they germinate at, because they're growing on deeper soils. So essentially they have water available for more of the time. So temperature is less of a constraint. These guys, the specialists, need to be able to, basically they need to know that they can germinate and grow rapidly And so they'll only germinate at a specific temperature, because water in these landscapes disappears fairly quickly. So they want to germinate as rapidly as they can. They will only germinate when their germination rate is optimal. But this whole trade-off between water and temperature led Sabashi, and by extension the rest of us, into this idea of, okay, well, for plants it's not just about temperature. They've got to choose between when can I be most thermally when is it most thermally optimal for me to germinate versus how much water is available and how long will it be available for. And so we kind of started playing around with the idea of these germination landscapes, combining mechanistic responses to temperature, such as we've seen so far, with mechanistic responses to water stress, which, as I've said previously, are sigmoidal. And so you can build mathematical equations that incorporate temperature responses and water stress responses, and you get these kind of, they're not really islands, they're sort of mountains or peninsulas of high germination capacity. And again, once we took the theory and started measuring germination capacity of different, this time acacia species, again, looking at the trade-off between range-restricted species and more widespread species, we had this idea that maybe you'd see narrower, tighter peninsulas for the range-restricted species, because they would only germinate under specific water stresses and specific temperatures. And we certainly managed to build the three-dimensional performance landscapes. And it sort of didn't play out exactly as we'd expected that it would do, because in this case, the species on the top row are the range-restricted species, and Wood maniorum seems to have what we'd expected, a sort of narrow, temperature-driven response, a sort of narrow peninsula. But Acacia carina was actually much less temperature constrained and much more water stress constrained. So, you know, it has a very short water stress response, but across a broad temperature range. And with the broadly distributed species, you see not the broad hump that we'd expected. But again, Assimilus and Exocarpoides both have their own specific thermal tolerance thresholds. So for the acacias, it was not quite such a cut and dried situation about how they responded, and it's because of the differing stresses, or the differing importance of those stresses, I should say, between temperature and water, and how the different species play those two stresses off. And I guess just to round this one out, this has some interesting conservation applications in its own right, because particularly in the study area we were looking at for these studies, for the Centre for Mine Site Restoration that I was working for, a lot of the landscape is flat and sort of sandy and loamy, like in the photo you see here. But scattered across this grey study area, there are these very small ironstone ridges, ironstone mountains, and you can kind of see one in the background of that photo there, where they're emergent ironstone hills. They're piles of iron ore, and they kind of emerge out of the surrounding plains. The soil type is different. There's a lot more topographic relief. the substrate is really cracked and broken, and there's a lot of water holding capacity in that substrate. And so each of these isolated ironstone ridges has a massive beta diversity turnover. They're little biodiversity hotspots, little islands in a sort of a sand plain sea. And each island has its own endemic species. And unfortunately, each island is also an economically valuable resource that is targeted for being dug up and sold as a mineral resource. And so across that study area, you have arrangements like this, where each of the points that you see there actually characterizes the entire global distribution of 4 species of short-range endemic Tetratheca. And those ridges are separated by maybe 100 kilometers maximum from the furthest north to the furthest south. So you can stand on top of these mountains and see the next mountain. And you can be surrounded by a population of one species of Tetratheca and know that the hill that you can see further in the distance has a different species that's found nowhere else. And so the question that we were confronted with is, well, how different can these species be? Because, okay, the microclimates on these hills are different to the surrounding plains, but the microclimate on a hill that I can see from here can't be that different to the microclimate of the hill that I'm standing on, surely. And so this is kind of where we went to, is how much insight can we get into the specific evolutionary requirements of these really unique species, and how does the hydrothermal physiology of them play out against a more traditional way of looking at niche envelopes. And so we went out there and we built, well, I did some biogeography mapping, calculated the soil water potentials and the soil temperatures for all of the known locations of these plants. And I should say, we have a very confident understanding of where every single individual of every single species is, because these species have been so extensively surveyed as part of the environmental protections associated with mining. So we're pretty comfortable we know where every individual is. And when you build niche envelope models of these things, yeah, each hill has about the same niche envelope. The microclimates are really not that different. And when you put all of the kernels overlapping each other, you get a combined hypervolume where pretty much every species is inhabiting the same microhabitat niche. So they should have the same stresses on them, and they should germinate in the same, with the same kind of hydrothermal capacities. But when we did the hydrothermal time modeling, we found that despite the similarity of their environments, that should say water potential, their thermal or their hydrothermal capacities are actually subtly different from each other. So you can see the 3 panels here, the three rows, represent water availability for different periods of time, 15, 30, or 60 days. And the 3 columns represent 3 of the 4 species of Tetratheca. And despite the fact that Tetratheca erubescens doesn't live in a particularly dry microclimate or a particularly hot microclimate, it actually is more hydrothermally robust than the other 2. It can germinate more quickly with a shorter water availability window. Tetratheca painteri, on the other hand, actually really struggles even if water is available for 60 days. So the takeaway message we had here is that once you have this mechanistic understanding of how temperature and water play out, you can really start to see subtleties that aren't inherently obvious just from looking at the environment of the species. And what that means for their biology, we are still exploring, and for their conservation, we're still exploring. And this is some preliminary work that is going to appear in the, or that we hope will appear in the upcoming special edition of Conservation Physiology on conservation physiology of plants. So if there are plant biologists out there, you've still got about a week to get your submissions in. I strongly encourage you to do so. It's going to be a fantastic edition of the journal. All right, I can see that Jody is asking me to get along. I'll close the blind here, and then B, I'll just finish off with some ideas about how I think this is going to go into the future. Because hydrothermal physiology is a really interesting idea for me. It immediately raises to me questions about isothermal physiology, because animals must also, some animals at least, must be constrained by an interaction between water availability or ionic balance and temperature. Off the top of my head, ones that occur to me as being really important are things like sea monkeys. Does everyone remember sea monkeys? The weird little toy thing that was sold when I was a kid? They're actually brine shrimp. Don't know why people wanted to grow brine shrimp as pets, but they did. And the thing about brine shrimp is that their eggs just roll around in the dust on the bottom of salt lakes and stuff for the better part of a year, or even longer. And then when that lake fills, all of a sudden the cysts hatch and there's brine shrimp everywhere, and they're reproducing, and you know, it's this kind of miracle of life thing where a barren sort of plain becomes verdant with life. I can hear David Attenborough like in my head quoting this kind of stuff. And there are species like this all over the world. This is a shield shrimp from Lake Eyre. And so these kinds of things, that there must be a trade-off between ionic balance and temperature that allows them to germinate and to grow the same way plants do. And the reason I think that's important is because these things form the basis of really vibrant food chains. This is an aerial photo of Pink Lake on Rottnest, and it's pink because it's filled with algae. And when the lake fills, it's a salt lake, and when it fills, all of the algae starts blooming, and brine shrimp hatch, and brine shrimp feed on the algae. And Pink Lake and a whole bunch of other salt lakes like it are hubs. It's a Ramsar wetland, I believe, because migratory birds then fly to these lakes. You know, they're part of the international migratory bird flyways, because they can come to these lakes and they can forage on the brine shrimp that hatch in these salt lakes and feed on the algae. So understanding how brine shrimp respond to hydrothermal stress actually has consequences that reach all the way from Perth to Alaska or Siberia. And as far as I know, no one's ever looked at isothermal performance norms in species like brine shrimp. And understanding these things is really important, because over the last 500 million years, there have been some really obvious rapid changes in temperature, in the global temperature. And every single one of them, bar one, has been associated with a mass extinction. The only mass extinction that wasn't associated with rapid temperature change was the Cretaceous extinction. And Larson, I think, nailed exactly why that happened. But all jokes aside, rapid temperature changes cause evolutionary problems, and they cause extinctions, or at least they're associated with extinctions. And today we have one of the most rapidly changing global environments in history. We've also lost half the world's agriculture, half the world's habitable land has been converted to agriculture. We've had a 60% decline in vertebrate populations, and our current rate of species loss is between 100 and 10,000 times the background rate. So we have a conservation problem, and traditional approaches to understanding conservation that don't capture the mechanistic capacity of organisms to respond to change, and don't capture the cascade that has through the ecosystem in cases like like the brine shrimp I was just talking about. If they can't respond to temperature, if they can no longer germinate in these salt lakes, a food source at the end of a migration path disappears for a lot of bird species. And if we don't understand that mechanism, these changes are happening too fast for us to approach conservation in the traditional adaptive management, I guess, paradigm. And that brings me on to where I want to close. This was some work that one of my master's students, Emily Tudor, produced earlier this year, because she took the idea of what if adaptive management incorporated physiology? And this is, I think, where we really need to get to. Her thesis statement, her rationale in producing this was to say that at every step of a traditional adaptive management loop, we can bring physiology into it. We can make physiological measurements. We can set goals with an understanding of physiology. And we can look at that at multiple different scales, at the scale of the organism, and at the scale of the landscape and the scale of the ecosystem. And if we don't do that, I don't think we're going to be able to develop the tools required to address the kind of conservation challenge that we've set for ourselves in the last 100 years. So I will end it there on that hopefully rousing and stirring note. I would just like to acknowledge all of the colleagues that I've made free of with this work. A lot of it was done by Sabashi, Emily Tudor, I've made reference to in that last slide. Colleagues at Curtin, colleagues at Kings Park, and of course the support of Kings Park, Parks and Wildlife, and the mineral extraction partners that supported much of this work. Under the auspices of the Australian Research Council's linkage and ITTC programs. I'll leave it there. Thank you very much.